Hello to all my dear students. Welcome once again on this amazing platform of Physics Wallah. Today we are going to start with the next lecture of molecular bases, that is lecture number 6. So far you have studied about the transcription mechanism. Now today we are going to see that how in case of eukaryotes post-transcriptional modifications are done so that heterogeneous RNA can be made ready for translation. Now beta, in case of eukaryotes, you know one thing, that both the processes, transcription and translation, are not coupled. There are two reasons behind it. Number one, in eukaryotes, transcription takes place in the nucleus of the cell, whereas translation has to take place in the cytoplasm. First reason. Second reason, modification. The RNA which is produced in eukaryote after transcription is yet not ready to be used for translation. There are certain levels of modification required so that the heterogeneous RNA produced in case of eukaryote with the help of RNA polymerase II can be made ready for translation. Modification is done in 3 levels. Number 1 is capping, number 2 is tailing, and number 3 is splicing. So let's start with the first post-transcriptional modification. But before that, let me write it. In eukaryotes, since RNA produced after transcription cannot be directly used for translation. Hence, 3 levels of modification are required. Hence, 3 levels of modifications are required. Number 1 is capping. Now suppose this is my RNA which is just obtained after transcription in case of eukaryotes. Right? Now this RNA is having some non-coding interrupting sequences also, which are called as interons. The proper coding sequences are called as exons. Such RNA is known as hnRNA or heterogeneous RNA. Correct? Now beta, the first step is capping. Capping means addition of 7-methyl guanosine at the 5' end with the help of enzyme guanyl transferase. Now, there is some enzyme called guanyl transferase present in the nucleus of the eukaryotic cell, which is responsible for the addition of 7-methyl guanosine. This extra cap is going to attach at the 5' end of the hnRNA. So addition of 7-methyl guanosine at the 5' end of the hnRNA with the help of enzyme guanyl transferase is known as capping. Now this 7-met-G at 5' end, why it is required to be done? Because now this RNA when which will undergo translation, so translation ka initiation position is recognized with the help of 7-met guanosine, so that correct translation can start during, once the RNA gets ready, then it should start from the correct position. It is the duty of 7-met guanosine to ensure. Hence, capping is very, very important for proper translation. Correct? So you can write this thing. Capping is required. Capping is must. Why? for proper translation later on. For proper translation, capping process is very, very important. Now, once 7-MET-G is attached beta at the 5' end, the next important modification is tailing. Tailing means some more residues has to be added at the 3' end of hnRNA. So, some free adenosine residues present in the nucleus of the cell, they are made to join a single strand with the help of a polymerase enzyme, poly-A polymerase, which is a template independent enzyme. Once that 200 to 300 adenosine residues are made to join to form a strand with the help of the enzyme poly-A polymerase, then that strand is later joined on the 3' end of the hnRNA. This is going to ensure the correct ending of translation in case of eukaryote. Right? So what is tailing? Tailing simply means, beta, addition of 200 to 300 adenosine residues. It ensures addition of 200 to 300 adenosine residues at the 3' end. Now, the chain of adenosine is produced with the help of enzyme with the help of template independent, because this enzyme is going to function independent of template. Right now you saw RNA polymerase, DNA polymerase. All these enzymes for functioning, they require a template. They require a base on which they read the strand and then they synthesize the new strand. But the first enzyme which I am going to tell you right now that functions in template independent manner is poly-A polymerase with the help of enzyme. In bracket you can write template independent. So with the help of the enzyme poly-A polymerase. Right? So now what is the condition after tailing? Suppose this is my hnRNA. Why? Because still exons and interons are present on the RNA together. Now in the beginning on the 5' end we have added 7-MET-G for correct beginning of the translation, and now at the 3' end we have added a chain of number of adenosine residues. This is known as polyadenylation. Capping is done, addition of 7-methylguanosine, and at last a polyadenyl chain is added on the 3' end, which is polyadenylation or tailing. Correct? Now, correct capping and tailing is done for correct beginning of translation and correct ending of translation. But translation can only happen when the RNA is homogeneous. But right now it is still heterogeneous. Why? Because coding sequences are interrupted by non-coding sequences. So for that, splicing is required to be done. Now what is splicing? Splicing, beta, just two terms you have to remember. Removal of, cutting off, removal of interons from hnRNA to obtain a homogeneous RNA only having coding regions. Right? First thing. Second thing, splicing is done with the help of some proteins like snRNA. snRNA is the major component of the splicing, which is along with the protein. So snRNA plus protein complexes is known as SNRPs. These SNRPs are required to cut the interons, and then once the interons are cut, This part will be removed, and then ligase enzyme will come to join the remaining fragments of the DNA, of the remaining fragments of the RNA to make a homogeneous strand. So second point is snRNA along with proteins called as SNRPs, they are required for splicing. What will happen? These SNRPs, they will cut the interons, they will remove the interons. Now fragments will be created, so that those fragments need to be joined. So at the end, after splicing is done, after removal is done, the remaining fragments of the mRNA, they are made to join to form a continuous strand of RNA having only coding regions. Once splicing is done, once tailing is done, once capping is done, your eukaryotic RNA is perfectly ready for translation. And right now it has also come out of the nucleus to the cytoplasm. So location is also correct, condition is also correct. Now this eukaryotic RNA can enter into translation. Clear? Now this RNA can be used to obtain proteins. Translation process we will do later on, but first important topic is beta genetic code. See, now RNA is ready. You are having all the types of RNA, mRNA, tRNA, rRNA. So with the help of transcription, you have produced all the type of RNA. Now all these three RNA, MRT, all these three together help in protein synthesis, correct? Of which messenger is carrying the information. I'm repeating once again, messenger RNA is carrying the information. It is carrying the information regarding that what protein has to be synthesized, number one. Number two, beta. Now, rRNA is responsible for the synthesis of ribosome that attaches on the mRNA for further protein synthesis. tRNA acts as an adapter between amino acid and mRNA. Now, mRNA is carrying the message. It is carrying the codons. But those codon will only be able to produce protein when right amino acid will come and bind to it. So bringing the right amino acid to the right codon is the duty of tRNA. So mRNA is the messenger. rRNA is making the ribosomal assemblies which will later on attach with the mRNA for protein synthesis, and tRNA is acting like a transfer or adapter RNA. So basically all the three types of RNA beta is going to help in protein synthesis. But first you have to understand that what is the information that mRNA is actually carrying. So that information of protein synthesis on mRNA is known as genetic code. So first you have to understand the message or the information that is being carried on mRNA. That means first you have to understand the concept of genetic code. First of all, the concept of genetic code was explained by scientist George Gamow. He was a mathematician, beta. He was a mathematician. He used to deal in with mathematical tools and mathematical operation. So basically, he used the concept of combination and permutation to realize or to understand the messages which are carried on the mRNA, and that was called as genetic code. Now, see, mRNA beta is a polymer of nucleotides. Nucleotides, there are 4 types of nucleotide possible, right? Now, suppose this is mRNA chain. It is messenger ribonucleic acid. That means this RNA is a polymer of nucleotide. How many nucleotides are possible? There are 4 nucleotides possible. Which are those? Adenosine, guanosine, cytosine and uracil in case of RNA, not thymine, you know that. Now, how many amino acids are there that need to be coded for protein synthesis, 4 nucleotides are there, and 20 amino acids need to be coded. So automatically George Gamow used combination and permutation method. He said that for coding 20 amino acid, the combination of these nucleotide on mRNA, suppose if it is 1, that means 1 nucleotide is coding for 1 amino acid, then only 4 amino acid could have been coded. 4 into 1 is 4 only, right? So what about the 16 amino acids? They would have not been coded by any codon. Okay, if genetic code would have been doublet, not singlet, then 4 fours are 16 amino acid could have been coded, but you know we have 20 amino acids to be coded. That means the only possibility is that genetic code should be triplet. 4 fours are 16, 16 fours are 64. That means 64 codons are possible on the mRNA chain, which is more than sufficient for coding 20 amino acid. Doublet is not possible. Singlet genetic code is not possible because they will not be sufficient enough to code the amino acid, as one codon can code for only one amino acid. So if 16 codons are possible, then 16 amino acids would only have been coded. So what about the remaining 4? Right? So singlet not possible, doublet not possible, triplet is more than sufficient. So obviously when triplet is already more than sufficient, then scientists will not think about quadruplet or pentagenetic code. They will stick to genetic code being triplet. That means 3 nucleotide on mRNA in a sequence forms one codon. And this one codon codes for one amino acid, which is responsible for producing different types of protein. Understood? Once again, I am repeating, according to George Gamow's combination permutation, genetic code has to be triplet. Beta, genetic code has to be triplet so that 64 codons can very easily code for 20 amino acids. According to which, if genetic code is triplet, then accordingly 3 nucleotide on mRNA chain will make one codon, and that one codon will code for one amino acid. This is unambiguous property. I'll tell you later on. So right now, have you all understood that why genetic code is triplet? So genetic code is simply triplet because this was just a thought of George Gamow, right? He did not perform any experiment in the lab. He was just a mathematician, so he used his combination permutation method to analyze that what should be the nature of genetic code, how the messages of mRNA should be read for protein synthesis. So according to him, on mRNA, the sequence of 3 nucleotide will read one amino acid. Why? Because there are 20 amino acids and 4 nucleotide. Singlet cannot be possible, doublet not possible, triplet genetic code is more than sufficient, because triplet genetic code means 64 codons can be coded, and like this we will get the answer that yes, 64 codons are more than sufficient for coding your 20 amino acids, right? However, later on this experiment, okay, first of all, just check this. This is a checkerboard which is showing you all the possible 64 codons. So how will you make this checkerboard? You know there are 64 codons. So and how many nucleotides are there? 4 nucleotides, right? And every codon will have how many position for nucleotide? 3. So this is how the checkerboard is prepared. I'll just prepare it for you also. Now just see. One, two, three, four. 4 nucleotides are there na. So we'll make 4 dabbas. Like this. Okay. Now, how many nucleotides in one codon? 3. So here you are going to write some nucleotides. This is one position of nucleotide. This is going to denote the one position of nucleotide. And here also we are going to denote the position of nucleotide. See, first, second, third. And how you are going to write the numbers? First, this is the first position. Here is the second position of the nucleotide in one codon. And here is the third position. Now, first nucleotide is U, C, A, G. Right? Then U, C, A, G. And in third beta, you are just going to write all the nucleotide in front of each block. U, C, A, G. U, C, A, G. U, C, A, G. See, like this. Right? Understood? Chalo. Now let's try to fill this checkerboard. So first position is U. Second position U. Third position U. Now again, first position U. Second position U. Third position C. First U. Same block U. A. U. U. Last position G. Okay, like this. Chalo. Now let's try to fill this block. A. First position. Second position A. Third position U. First A. A. C. A. G. Right? Now let's try to fill this position. G. C. U. G. C. G first, second C, third A, fourth G, C, G. This is how you are going to fill all the codon 64. Just remember that this position is telling you the first position of the nucleotide. The top one is telling you the second position of the nucleotide, and on this side you're going to mention all the nucleotide in the sequence UCAG only, and this is going to tell you the third position of the nucleotide in one codon. And like this you can fill the whole block. You can just check it out. See, first position UUU, second UUC, third UUA, fourth UUG. Right? So this is how whole or all 64 codons can be filled. Now, this experimental proof, the experimental proof of genetic code being triplet was given by Meti Nirenberg and Khurana as well. So, okay, the experimental proof. Now see, this fellow, George Gamow, as I already told you, he was just a mathematician. He simply used his combinations and permutation to explain that genetic code is triplet. Now, scientists like Nirenberg and Meti, they just got the idea that yes, genetic code can be triplet. But being scientists, they said, okay, bhai, George Gamow just calculated with the help of some formulas, with the help of some logic. But is there any experimental proof that can be the genetic code triplet? So later on, all these scientists, Nirenberg, Meti, and yes, this fellow Khurana, who's an Indian origin scientist, they performed the experiment to prove that George Gamow is whether saying correct or not. So the experimental verification of genetic code being triplet was given by Nirenberg and his colleagues. Nirenberg and his colleagues, they performed the experiment to prove that whether Gemo is saying right or not. So for this, first of all, they discovered a template independent enzyme called polynucleotide phosphorylase, also called as Severo-Ochoa. And they took a test tube. In this test tube, they added number of nucleotides, but all were uracil based. So they added n number of nucleotides of uracil. But whatever the number was, it was in the multiples of 3. Because somewhere Nirenberg, they were having the idea that genetic code can be triplet. So whatever nucleotides, maybe uracil, they added in the test tube, n number, but that number of nucleotides added in the test tube was the multiple of 3. Apart from adding these nucleotides, then they added an enzyme which is Severo-Ochoa. Severo-Ochoa, basically beta, it is a template independent enzyme. That means it functions independent of template. It can simply join all the Severo-Ochoa, which is also known as polynucleotide phosphorylase. Right? So this enzyme is going to function independent of the template. This enzyme, what is the role of the enzyme? Okay, apart from this, some magnesium ions, energy, many more things were provided which is required for both translation process. Now this enzyme, Severo-Ochoa, in independent, template independent manner, it will pick up all the free uracils added in the test tube to make a RNA chain. Right? So this enzyme is going to convert the free nucleotides in the test tube in a form of a single stranded structure with the polarity 5' to 3'. So first step in the test tube is going to be the synthesis of this chain. Later on, this chain, with the help of other substances provided, will undergo translation process, protein synthesis. So after protein synthesis, what this fellow Nirenberg, इन्होंने क्या observe किया, that after the whole thing got completed in the test tube, they obtained a polypeptide of 1, 2, 3, 4 phenylalanines. They obtained a homopolymer chain of protein phenylalanine. So with this experiment, he concluded that definitely genetic code is triplet. Why? Because he added nucleotides in multiple of 3. So this is if genetic code is triplet truly, then if he added 3, 6, 9, 12. If he added 12 nucleotides, then surely he should get a polypeptide chain of 4 amino acids of 4 protein. So is it not true? See, he added 12 nucleotides in multiple of 3. So he obtained 4 polypeptide, 4 amino acid chain. Hence, genetic code is triplet. First thing, that means each 3 nucleotide is making 1 codon, and that 1 codon triple U is coding for phenylalanine, and hence the chain of phenylalanine is being obtained. Homopolymer, why? Because all the proteins are of same nature. So homopolymer. Second, none of the nucleotide is overlapping. You can see 3 nucleotide is making 1 codon. So those 3 nucleotides are not becoming the part of another set of codon. Every nucleotide is completely donating itself to make 1 codon only. That means genetic code is non-overlapping. We understood. Third point. We understood that obviously it is unambiguous because all the time triple U is coding for phenylalanine. That means 1 codon will always code for 1 amino acid only. If this would have not been there, ठीक है, यहाँ पे triple U coded for phenylalanine, then next triple U should code for another amino acid. But no, it is not happening. Every time triple U is coming, every time it is coding for phenylalanine. That means it is specific, it is unambiguous. One codon will only code for one amino acid. And there is no commas, there is no, this is the beginning point, this is the end point. So once the process starts, there is no breakage, there is no stopping of the process till the time end point does not come. So there is no comma, there is no punctuation, less genetic code. Once it starts, it will only going to end at the end point, nowhere in the middle if all the conditions are properly provided. So that means genetic code is read in a contiguous, in a continuation manner without any pause or comma. So these were some properties which were analyzed with the help of the experiment performed by Nirenberg, and that you are going to write in the salient features of genetic code. So first salient feature says that genetic code is triplet. This is now proved. The thought of genetic code being triplet was given by Gemo. Later on, it was proved by Nirenberg and his colleagues. So genetic code is triplet. This is universal. Second, genetic code is non-overlapping. Right? That means 3 nucleotides which are donating themselves to make one codon, then they will not participate in another codon. Right? Third, comma less or punctuation less genetic code. Once the process will start, it is not going to end or it is not going to stop in mid. The process of translation will only stop once the end point come. That means genetic code is read in a continuation manner. Fourth property, unambiguity. Beta, unambiguity means what? Unambiguity means at a time one codon will always code for the same amino acid, will code for same amino acid. Like for example, triple U always code for phenylalanine. Right now you have seen in the experiment of Nirenberg also. Next point. See, there are how many total codons? Total codons possible is 64. Why? Because our genetic code is triplet. Right? Out of 64 codons, beta, 61 codons are active, which can help in the coding of amino acid. Why? Because 3 codons, that is UA, UAA, UAG and UGA. These 3 are nonsense codon or termination codons which simply end the process of translation. They do not code for any amino acid. So these 3 are beta nonsense or stop codon. So why they are nonsense or stop codon? Because they do not code for any amino acid. They simply stop the process of transcription. They stop the process of translation. Clear? Understood? Next. So these are basically called as termination codons. Now, apart from termination, there is one codon which always initiate the process of translation, be it in prokaryote or in eukaryote, which is AUG. So next is initiation codon. These are universal thing with few exceptions. Exceptions we are not going to do. Few exceptions can be there, but we are not concerned about the exceptions. You should know the main concept. So initiation codon is AUG, which codes for methionine. So first amino acid in every chain of RNA has to be methionine. Understood? Next, genetic code beta shows degeneracy. Degeneracy means, see, One codon will code for one amino acid only, but one amino acid can be coded by more than one codon. This is degeneracy. One amino acid can be coded by more than one codon. Like for example, triple U codes for phenylalanine, right? UUC also codes for phenylalanine. So there are two different codons but coding for same phenylalanine. Check this checkerboard. See, go back and check. Triple U phenylalanine, UUC also phenylalanine. Now one more, UUA leucine, UUG also leucine, CUU also leucine, CUC also leucine. So many leucine are coded by different codons. One codon, one amino acid, unambiguity. One amino acid, many codon. This is degeneracy. These two are very important features of genetic code. Degeneracy, unambiguity, being triplet, comma-less, non-overlapping, initiation codon, termination codon. All these are the beta. Yes, these all are the properties of genetic code which you need to remember. I hope all the concept related to genetic code and their features is clear. Exceptions need not to do. Now, before you start with the main process of translation, you need to understand the structure of tRNA also, because apart from mRNA and rRNA, the third important RNA that is also involved in the process of translation is tRNA. T stands for transfer, right? Transfer, also called as adapter, whose function is to add specific amino acid to the specific codon on the mRNA, right? Now, codons are 64. Amino acids are 20. Number of tRNA is according to the number of amino acid. tRNAs are specific to specific amino acid only. First thing. Now, two structures of tRNA were proposed. One is a two-dimensional structure which was given by scientist Holly. That structure is there in your syllabus. And another structure is three-dimensional proposed by Kim. Three-dimensional is inverted L shape. Two-dimensional is clover leaf-like. So this clover leaf-like structure we have to study. Two structures. 2D by Holly, which is clover leaf-like. This is in your syllabus. Otherwise, one more structure, three-dimensional structure was given by scientist Kim, which is inverted L-shaped. Right? But now we have to see this simple structure of tRNA, which is which is clover leaf-like. Now, again, our tRNA is also single-stranded. So, single-stranded means 5' to 3', but it is not like a chain. Rather, it is some twisted to form arms and loops. So, this is a 5' end having phosphate. Then this 5' end of RNA turns to form arm, then loop, then again turn, arm, loop, arm, loop, and then it ends over here. 3' end hydroxyl group is there, followed by CCA nucleotides from 3' end below to it, CCA. This is how the structure of tRNA appears to be. It is not straight like this is simply mRNA. A straight single-stranded structure, but tRNA is slightly twisted to form arms and loops. Right? Now, this first arm, see, this is the first arm. From 5' end, this is second arm. From 5' end, this is third arm. This is fourth arm, and this is variable arm, extra variable. There are names to every arm and there is function associated with every arm and loop. So these are the loops. first arm is not having any loop and variable arm is also not having any loop. Clear? Now, first arm is known as amino acid binding. Here on this first arm, amino acid is going to bind. So it is known as amino acid binding arm. Second arm is known as dihydroxyuridine arm, DHU. Dihydroxyuridine arm. Third arm beta, the one which you see over here opposite to first, is known as anticodon arm with anticodon loop. And the fourth which you see is known as T-Cy-C arm or ribosomal binding arm with a loop, right? Now, arms are beta having paired nitrogenous bases. They're having paired, n number of paired nitrogenous bases. Even being single-stranded, but they have paired nitrogenous bases. However, the loops are having unpaired nitrogenous bases. They are fixed in case of anticodon loop, which is 7. 1, 2, 3, 4, 5, 6, 7. Here also 7 fixed. 3, 4, 5, 6, 7. Right? So arms are having paired nitrogenous bases. Loops are having unpaired nitrogenous bases. Right? Main concern is with the first arm and the third arm. The first arm is responsible for binding to specific amino acid, and anticodon, this I need to explain you, which is very, very important. Now, in case of anticodon loop, beta, the 3 nucleotide, in case of anticodon loop, let me take one more page. See, this is the structure, right? This is how it is. Here are 3 anticodons. There are 3 nucleotides which are called as anticodon. Anticodon means opposite to the, complementary to the codon. Now suppose this is my mRNA chain, single stranded 5' to 3'. Now suppose mRNA is having A, U, G as nucleotide. This is one codon on mRNA. Now the tRNA which is going to bind to this codon on mRNA should have anticodons on its anticodon loop, which is going to be AUAC. So anticodon is that loop which is having anticodon with respect to the codon on the mRNA. Now here, with the help of hydrogen bond, the tRNA is going to attach on the mRNA. Correct? Now, AUG codes for methionine. So this tRNA is going to allow the binding of methionine on its first arm. So what this tRNA is doing, beta? See, this is tRNA is acting like an adapter. It is joining amino acid with the specific codon on the mRNA. With the help of its anticodon loop, anticodon arm or anticodon loop, and amino acid binding loop. The anticodons, they are complementary to the codon present on mRNA, and whatever amino acid is brought is according to the codon present on the mRNA. This is how tRNA functions, and tRNAs are specific to specific amino acid. I hope you all have understood the structure of tRNA. If yes, then there are certain questions for you to solve. Please take them as homework. And yes, with this, we come to the end of a simple but easy and very crisp lecture on genetic code, structure of tRNA, and yes, of course, post-transcriptional modifications in eukaryote. 3 major topics today you have covered. In your next class, we will simply deal with translation. And again, I will give you the hint on the structure of tRNA. So till then, keep watching the lectures. Bye-bye everyone. Take care.